Hi, I'm Stan Nelson. I'm professor in the Department of Human Genetics, and I'm co-director of the Center for Duchenne Muscular Dystrophy. The next set of lectures are about the genetic discovery of the molecular basis and the mutations that cause Duchenne muscular dystrophy. It actually creates a spectacular tale and creates a lot of novel creative thoughts that went into one of the very first human disease genes positionally cloned in history. So for this process, what I want to do is put you in the mindset of where we were back in the early 80s. So we knew that Duchenne muscular dystrophy was an X-linked recessive disorder because of how it was transmitted in families. We also knew roughly that it was one of the most common genetic diseases of childhood, about 1 in 3,500 to 1 in 5,000. The gene is called DMD. We know that now. We know the protein, which is called dystrophin. And the gene was really identified in 1986. It was co-discovered, it was identified, the first mutations were observed by Lou Kunkel and Ron Wharton, simultaneously using very different approaches to winnow in on that exact location. And Lou's team was able to figure out exactly what the mutations were and what dystrophin, the protein, looked like at the end of the day. So one of the things I'd start you off with here is when we're starting in 1980s, we knew where the gene was to a 5% accuracy. So out of the 100% of the genome, we knew it had to be on the X chromosome. So that's about 1/20th of the genome. So that's pretty substantial in terms of reducing where the scientists were going to look at that day and age. So if you think about a Punnett Square in terms of the number of kids that are affected from a carrier mom, indicated across the top, or an unaffected father, indicated on the left, that 25% chance that that couple will have an affected child, and that affected child will almost always be a male child. There's a 25% chance that that couple will have a carrier child, and that carrier child will always be a female. So a few of the carrier females are indeed affected or manifest substantial disease issues, but typically later on in life. And then there's about a 50% chance that the mutant X chromosome was not transmitted to either a male offspring or a female offspring. So in total, about 75% of the offspring from such a couple were not affected. But we didn't have good ways to assess whether a female was affected, and we actually didn't even have great mechanisms necessarily to tell that a female was a carrier without a genetic test. So one of the first things to be faced with was if you're faced with this family where you have an unaffected father, an unaffected mother, the mother may or may not be a carrier, and there's an affected child in the family, how do you counsel that family for the next and subsequent kids? It was very difficult at the time. Further things happen is, I just told you that Duchenne muscular dystrophy is an X-linked recessive disease, but this is data from a paper published in 1980 indicating a Duchenne muscular dystrophy being inherited in an autosomal recessive pattern in 13 different families, identifying 31 cases. What's going on here? Is it autosomal recessive, or is it X-linked recessive? And the answer ends up being that this is a disease that looks a lot like Duchenne muscular dystrophy, but happens equally in males and females, and has an has an autosomal recessive inheritance pattern. And it ends up being a rare form of limb-girdle muscular dystrophy. We now know, because of the spectacular explosion of being able to do linkage, predominantly in the 1990s, followed by very detailed, focused cloning experiments and identification of specific mutations, that there are dozens and dozens of forms of molecularly defined limb-girdle muscular dystrophies, of which Duchenne muscular dystrophy is one type. These other types, particularly of the rare recessive forms, are generally less common in the population and are generally due to one mutation from one parent, the other mutation from the other parent, and the affected child. So the bolded area is limb-girdle muscular dystrophy 2C, which is caused by gamma sarcoglycan, which is the answer to the Tunisian family. So that got worked out, and understood as really a very fundamentally different muscular dystrophy. I'm highlighting here as well limb-girdle muscular dystrophy 2K due to POMT1 mutations, partly because that will be towards the end of the lecture where we talk about the immense genetic heterogeneity of muscular dystrophies, and we're still not at the end of solving all muscular dystrophies genetically. So one of the benefits of going after what the precise molecular basis of Duchenne muscular dystrophy is, using a positional genetic strategy, is that you don't have to take guesses at what the disease is caused by. You can use the positioning in the genome, you can go after the gene, and the genetics tell you that this gene causes, or the lack of this protein causes a specific disease. So before that, we're at the liberty of making all sorts of assumptions. So this is a Duchenne hypothesis before gene discovery, published in Muscle and Nerve, and it's Duchenne is being considered as a disturbance of nerve-muscle interactions. Discovering what the cause of Duchenne is genetically revealed that indeed nerve-muscle disturbance is not the cause of Duchenne muscular dystrophy. Here's a paper published many years earlier in Science, one of the leading journals, indicating that Duchenne muscular dystrophy is the result of functional ischemia because you could reproduce a lot of the disease manifestations by inducing functional ischemia in model organisms. Again, the discovery of the Duchenne muscular dystrophy gene indicates that this is not the proximate primary cause of Duchenne muscular dystrophy. So what's happened in this last 20, 30 years is genetics has been used, and particularly positional genetics has been used to unravel the molecular basis of thousands of rare Mendelian genetic diseases, and that these have been largely positionally cloned, where we used information about family structure, linkage, positioning the gene in the genome, and then identifying a sequence difference, a mutation that moves consistently through the family and is completely associated with the disease, either in a recessive form or a dominant form. And this allows us to winnow in on the disease gene without understanding the molecular basis of the disease first. That is, we're not taking a candidate approach, we're allowing the power of human genetics to find the disease. And this has been immensely powerful, leading to thousands and thousands of different discoveries. An important part of this is linkage analysis. So coming up with markers, DNA markers that are codominant, which simply means that we can see both alleles, the mother's allele and the father's allele in a given individual, and we can observe them both. And you can observe each one of those markers either being transmitted or not being transmitted to every offspring. So this, if a marker is very close to a disease, the disease and the marker always co-segregate, and that's linkage. So if a disease and a marker were right next to each other, there'd never be a recombination, and they'd always go together. If a disease gene and a marker were 50 centimorgans away, 50 megabases roughly in the human genome, or on another chromosome, they'd be co-inherited 50% of the time, which is just chance, and therefore unlinked. So you can use that very powerfully, and that has been used many, many times over to identify many of the different disease genes, to first find a position in the genome, clone all the genes in that region, and then identify the causal mutations. This is what this looks like pictorially. This is an example of an autosomal dominant trait where the disease and the gene is being passed and transmitted to about half the offsprings in each individual generation. A typical way that this disease gene would have been solved is by doing linkage mapping within many such families like this, where you're observing multiple different transmissions and can show a non-random co-inheritance of the allele of the codominant DNA marker, which early on was restriction fragment length polymorphisms, which I'll talk about in just a second. And that gets you to a crude location in the genome, typically millions of base pairs, where one then typically goes about either finding more families to winnow in on the location more, cloning out all the genes and identifying the genes in that area, and then sequencing every single one of those disease genes, or every one of those genes, with the idea being one of them must be the disease gene, and you can get at that at the end of the day. An alternate approach, so that was one approach that was actually used to indeed identify where the Duchenne gene was on the X chromosome, was using linkage. There's also other surprising things that happen in human genetics and in humans that create the diversity of humans that are on the planet. So what was observed in the 60s were a group of kids who had unusual X chromosome abnormalities. So it might be X in Turner's individual, it might be an X chromosome translocated to another autosome, and some of those girls actually manifested Duchenne muscular dystrophy, which was sufficiently unusual to beg people to look very carefully at these females for what that reason is and what was causing that. So in the 70s, when some of the cytogenetic tools were improved, there were multiple different females who had a translocation from the X chromosome, where we now know the Duchenne muscular dystrophy gene is, and where we knew it was just from being an autosomal recessive, from being an X-linked recessive disease, to multiple different autosomes. So that translocation was observed over and over again, different autosomes, but on the X chromosome, for each one of those girls, it always broke into the X chromosome at XP21. And all 5 of those females had Duchenne muscular dystrophy. The reason for this is an interesting molecular issue. That is, in females, typically one X chromosome is turned off randomly in every single cell, so that you have expression from either the paternal X chromosome or the maternal X chromosome only, one of those two, and it's random across all your cells. So it's random X inactivation. When one of the X chromosomes is mutant and is translocated, the other X chromosome, the normal X chromosome, is the chromosome that's always turned off. So these girls all manifested Duchenne muscular dystrophy because there was an alternate genetic mechanism for turning off the normal X chromosome, which didn't have a DMD mutation. This led to a very powerful observation that indeed the DMD gene was localized cytogenetically to this location of XP21. That's pretty big, that's tens of megabases big, but still gives you a pretty good location. So now we've went down from 5% of the genome down to just 1% of the genome, roughly. So we know roughly where it is, but there could be hundreds of genes within that interval, so it hardly tells you what the exact molecular cause is. At this same time, restriction fragment length polymorphisms were being suggested as possible codominant markers. Now we're back in 1980, where this idea was first proposed. And the idea is that if you had a polymorphism within a restriction enzyme site, so your DNA difference between 2 different humans disrupts a restriction enzyme site, when you make a southern blot of a restriction enzyme digested genomic DNA, it will look different in different humans, and that will be transmitted generation to generation in a very consistent, very easy to recognize way. So on the right-hand side of this slide are 2 different people with 2 different restriction fragment patterns, and this is due to a DNA polymorphism within the restriction enzyme site. So we can use these powerfully to do linkage in general. And as a matter of fact, in 1980, a very powerful paper demonstrating that you could find these codominant markers was published by Botstein and colleagues. And by 1982, Jim Murray and Kay Davies identified a marker on the X chromosome that was a restriction fragment length polymorphism that was about 10 centimorgans away from the Duchenne phenotype, so that there was about a 10% chance of recombination between the allele that you could now track and where the Duchenne gene was. So this actually provided for the first time a DNA diagnostic that was of some utility for determining what the chances were of a second child. However, only 29% of all females were heterozygous for this DNA variant. So if you weren't heterozygous for it, it wasn't useful for determining whether the disease was on one chromosome or the other. So 70% of women, this was no use for. So it was a very imperfect DNA diagnostic, but was indeed of some utility during this period. There were 2 different approaches taken to winnow down and get to the race and get to the goal of finding what the DMD gene is. What's the RNA and what are the mutations? And then what's the protein that's made at the end of the day? So one approach was taken by Kunkel. And he took a very clever idea that he'd used previously for cloning out specific different regions of the genome. And he took a specific DNA subtraction tool approach to identify probes on the X chromosome that could then be used as tools for trying to find the Duchenne gene. An alternate approach was used by Ron Wharton, and one of the kids had a translocation of XP21, so they reasoned that on the X chromosome, that break occurred right within the Duchenne gene, so that it broke the Duchenne gene. It's a reasonable supposition. The other end was on chromosome 21, and in chromosome 21, he knew that there was a ribosomal gene cluster, and he could get probes for ribosomal gene clusters. So what he reasoned was that if he probed a library from this person with ribosomal gene probes, he would eventually find a clone that starts in the ribosomal gene cluster but ends over an XP21, and that bit of DNA would indeed be mapping within the region that's mutated in Duchenne muscular dystrophy. The one part that I haven't told you yet is people had already observed deletions as well within the X chromosome, and deletions around XP21, and there were a few rare patients who had multi-gene deletions and had not just Duchenne, but other diseases, and they were cytogenetically visible, so they were fairly big deletions. So people already knew that big deletions could cause Duchenne muscular dystrophy, just didn't tell you what the gene was. Exactly. So both of them published papers identifying where the precise location was of the Duchenne gene, and actually both were able to make DNA probes that could detect DNA deletions in a small fraction of the patients, but again, the gene itself had not been identified. So these are deletion-type mutations that they could see, but not necessarily know exactly what the gene was. So they're a step closer. They're both right within the gene at this point in 1985. Kunkel's approach is what we're going to focus on. It's a clever strategy, and it ultimately went forward with identifying the full set of all exons and the full cDNA and the full protein that's encoded that leads to Duchenne muscular dystrophy. So that's what we're going to focus on now. What Kunkel did was he took a clever approach of a DNA subtraction strategy. So he took a patient BB, who has XY chromosome. He's a male. He has Duchenne muscular dystrophy, and he actually has a couple of other diseases as well. He has a big deletion. That big deletion has to include the DMD gene, as what Kunkel had reasoned. So it's missing completely, and it's big because it's affecting multiple genes. So what he did was he took that DNA and he made a vast amount of it, and he created blunt ends so that they wouldn't be compatible with cloning into a sticky end vector. So it's a low molecular biology trick that he was using at the time, and he puts a vast amount of that type of DNA in. And the idea is he then takes a person with 4 X chromosomes and 1 Y chromosome, so a lot of X chromosomes, with the idea being he cuts that library with sticky ends that can clone in. So only if they self-anneal back to each other can they be cloned successfully into the vector. So what he's looking for are DNA sequences that are missing from the XY genome here and present in the XXXXY genome. And he successfully does that, and does that very spectacularly well. The process required mixing these two genomic DNAs, a lot of the XY with the deletion and a small amount of the other. Together, you let them hybridize for a very long time, 5 days actually, and then you take what's left and successfully annealed as double-stranded DNA that now has the appropriate sticky end site and clone that into a vector that can only clone that out. And he indeed found a number of different clones that map to this region. One of the clones, PERT87 probe, was missing in the genomic DNA from 6.5% of all Duchenne boys. He knew he was within the gene with this one small plasmid probe. So that's fantastic, and lets him start figuring out where he's going to be at. So in this slide, the deletion is indicated here and indicating that indeed the PERT87 in lane 6 is missing from this individual, whereas that probe is more highly abundant, the PERT87, in the individual who has XXXXY. So we're very close or within the actual disease locus, but he doesn't know where he is yet. He doesn't know where the gene is. He doesn't know where the RNA is being encoded. And he doesn't realize how big of a problem he's going to land in, because he just landed in the largest known human gene. So at actually 0.1% of the human genome encodes the dystrophin gene. It's encoded over a 2.5 million base interval. And it's really rather remarkable how quickly they were able to work a lot of these issues out. The PERT87 landed them in the right spot, but they didn't know whether to go right towards the centromere or left towards the other telomere on the P arm of the X chromosome. So they had to just start walking and kind of randomly building clone libraries that got them further and further away from PERT87. And this is a method that's called chromosome walking. And the idea is you have a library of the X chromosome, and you probe with the PERT87 probe, and then identify other hybridizing molecules and use a restriction fragment map to identify that you've got some new genomic DNA sequences in a new clone, and then you clone bits from that, and then you use that to probe again the same library and pull the next clone over, and you keep iteratively doing this until you've built a restriction fragment map of the interval, and you know that you have clones spanning the interval. So Lou Kunkel obviously had a talented group of postdocs and graduate students working on this project, and very quickly they identified a whole set of different lambda clones marching across this interval. That led them to identify potential candidate cDNAs, because somewhere within this interval they should have the gene. So this is actually an example of about 200,000 bases of genomic DNA cloned into specific fragments with an exact linkage map. They're only 1/10 of the way done of actually finding the whole Duchenne gene at the end of the day. So pretty remarkable how quickly they end up finishing this up. But this was sufficient to start saying, I might have some cDNAs from these clones and from sequencing, finding open reading frames and saying that I might have an actual cDNA from looking at the genomic DNA. So this is one of those probes from PERT, it's PERT87-25, and PERT87-25 is a proposed cDNA probe. So to prove that it's detecting a gene, they then probe a northern blot. So take all the RNA, run it out on blot so it's size separated, and then probe it. And indeed what they were able to see was that the fetal muscle samples detected an RNA transcript that was about 16,000 bases, is what they reported here. Ends up being about 14,000 bases later. And that probe is therefore an mRNA. If that probe is detecting a cDNA or an mRNA that is at least expressed in muscle. And it detects one really big RNA. So that was exciting, and indeed they might have the gene in hand. And that was sufficient then to start going and figuring about that 116,000 base clone. Could that indeed be the dystrophin gene? Could it be the causal gene that leads to Duchenne muscular dystrophy? They were able to show here in the A plot, the BB patient is lane number 3, and indeed there's no hybridization with the probe to that person because he's deleted in this interval, and the other hybridizations are consistent with this being the appropriate X-linked probe for this cDNA. That led them to therefore clone out the complete cDNA sequence of this 16 kb detected mRNA, and this is represented here. It's present within multiple different independent clones, and it ends up being 14,000 base open reading frame that is indeed the Duchenne muscular dystrophy gene, DMD, that encodes dystrophin. So this process, they now have the full cDNA sequence. That's spectacular, and it allows them to start looking at what are the molecular sets of mutations that occur in Duchenne muscular dystrophy. So very quickly, with having all of the sequence of the cDNA, even though it's spread out over 2.5 million bases of genomic DNA, they could start looking at what's the spectrum of mutations. So very quickly, over 1,000 patients were assessed because it's one of the most common human diseases. Lots of patients had been identified. And indeed, most of the mutations were deletion-type mutations and could be quickly ascribed. And this immediately has an impact of you can now have very specific molecular diagnostic tools for a given individual patient. You know their exact mutation in the boy who's affected. That allows you to do a DNA testing in the mother to see whether she's a carrier and give families far more precise data than was able to be done immediately before this paper was published and the data and the tools were available. It also allowed, once you had the cDNA in hand, to start creating protein probes that allowed Lou Kunkel's group to identify the dystrophin protein, the protein that's encoded by the 14,000 base fragment. This happened in the following year, and indeed they were able to identify a huge protein, 427,000 Dalton protein, and were able to identify that it was present within the membrane fraction, and indeed start to dissect why this missing protein leads to Duchenne muscular dystrophy for the first time. And we've had an explosion of knowledge about that over the course of this last 30 years. From a genetic point of view, I'll point out the dystrophin protein drawn here pictorially is all of the darker blue ovals, I guess those are. And it's a stretch, a very long, largely filament-like protein that at the N-terminal domain binds to actin, and at the C-terminal domain binds to beta-dystroglycan in the muscle membrane. What happened from identifying Duchenne muscular dystrophy protein was it led to the identification of all of the components of a multi-protein complex called the dystrophin-associated glycoprotein complex, and many mutations within those components lead to many different other forms of muscular dystrophy. And now we actually understand that alpha dystroglycan, which is the purple barbell-shaped protein, is heavily glycosylated, and defects in just the glycosylation of that one protein. The protein can be there, but just one of the glycosylation defects leads to profound manifestations in humans, and there's now over a dozen different forms of disease genes that are identified that just lead to alpha dystroglycan glycosylation defects as rare but very significant forms of muscular dystrophy. So in this section of the lecture, what I want to get across to you is identifying the Duchenne muscular dystrophy gene used a positional cloning strategy years ago that was quite creative and led to a spectacularly important part of muscle function. Understanding what the dystrophin mutation has allowed us to better understand all the other proteins that are involved in muscle function. We're still not done with that process, but to give you a sense of where we're going to go now is gamma sarcoglycan, which is one of the yellow ovals in the middle of the muscle membrane, was the mutation that used to be ascribed to the DMD phenotype that was autosomal recessive in Tunisia. It's now known as gamma sarcoglycanopathy, and is its own distinct disease with its own distinct molecular basis, genetic counseling, etc. Alpha dystroglycan, the dumbbell-shaped thing, has multiple different genes that are defective that lead to its dysfunction as well. So by understanding any one of these components, we move to understand better muscle cell physiology, but also we're better enabled to find all the other mutations that are leading to the many, many, many different forms of muscular dystrophy. Thanks for your attention.